Thanks for joining this talk in Nanoscientific Symposium 2020. My name is Myunghoon Choi. This talk is on behalf of Professor Lane Baker. I'm going to talk about what we can learn from the studies of a precision synthesized gold nanoparticle catalyst by utilizing scanning probe microscopy. Before start, shortly introducing myself, previously I used to work in Park Systems as AFM and SECCM application engineer for several years. To study further, I have joined the Baker Lab, Indiana University in US. It is a fourth year of a PhD study this year. This talk will present two recent studies about precision synthesized nanoparticle catalyst. In the first study, individual electrocatalytic activity of the synthesized nanoparticles were examined by an SPM called Scanning Electrochemical Cell Microscopy, shortly SECCM. The second study is about characterization CTAB adsorbate on the surface of a synthesized gold nanoparticle. This whole story of today's talk is from a research collaboration with the Professor Ye group, who provided the precision synthesized nanoparticle. Synthesized nanoparticle catalysts from materials chemistry provide new opportunities to understand and tune the structure-function relationship for developing higher efficiency catalysts. Such a synthesized nanoparticle has different physical, chemical, and electronic properties depending on the structure and corresponding exposed low index planes. For example, gold nanocube exposes FCC low index planes 100 facet, while gold octahedron exposes FCC 111 facets. So one basic difference between the two, FCC 100 has a lower surface atom density than FCC 111. SECCM is an optimized tool for nanoscale electrochemical analysis introduced by Unwin Group in 2010. SECCM utilizes a droplet at the tip of a sub-micrometer dual barrel pipette as an electrochemical cell. This tiny electrochemical cell contacts the conductive sample surface and performs cyclic voltammetry to record electrocatalytic response of a local area. SECCM measures a series of cyclic voltammograms in massive numbers and create a spatially resolved electrochemical activity map. In Baker Group, SECCM was constructed on an AFM and SECCM platform over Park Systems instrument by equipping a custom-made potentiostat, probe positioning system, and humidity gas chamber, and relative humidity and temperature monitoring system. Figures here are representative SECCM results acquired from precision synthesized nanoparticle catalyst, in which the electrocatalytic activity of activity map of a hydrogen evolution reaction is present for gold nanocube and gold octahedron. So gold nanoparticles were drop cast on the glassy carbon substrate for SECCM measurement, and cyclic voltammetry was performed in each pixels of a map with 100 millimolar perchloric acid. So figure A is the SECCM CV map acquired at negative 0.95 volt versus silver chloride or gold nanocubes. So which shows a higher electrocatalytic activity of nanoparticles than compared to a glass carbon substrate shown as bright colored pixels. So the electrocatalytic response signal acquired from a single nanoparticle is marked as a green circle in figure A and B. So figure B shows a corresponding SEM image taken from the same area where SECCM map was taken. So by correlating the two, the cyclic voltammogram shown in figure C was chosen, which represent specific electrocatalytic response from a single particle only. So figure E to H shows the same SECCM measurement result for gold nano octahedron. For statistical analysis, 101 CVs from individual single gold nanocubes and 99 CVs of gold octahedrons were collected. The plots shown here, the average cyclic voltammogram of each with a standard deviation. The inset plot is without standard deviation mark. So red plot represents the electrocatalytic response of gold nanocubes, and blue plot represents gold octahedron. So the current magnitude increases for both nanoparticles about at negative 0.6 volts. As the potential becomes higher, the current magnitude of gold nanocubes increases rapidly than the one of gold octahedron, which clearly proves that gold nanotubes of facet 100 has higher electrocatalytic activity of a hydrogen evolution reaction than gold octahedrons. So this result supports our previous report of density functional theory DFT calculation about hydrogen evolution reaction activity on gold surface, which is suggested that gold 100 surface can be superior for hydrogen evolution reaction compared to gold surface 111 facet due to the more negative hydrogen adsorption energy of gold 100 facet. In this second study, gold nanocube was utilized as a model system due to the simple and symmetric geometry in nanoscale. And in the platform of model system of nanocubes, the CTAB adsorbed on the gold nanocubes was characterized with various microscopies. One well-known method to synthesize nanoparticle catalyst is seed-mediated method. So in this synthesis method, the crystal growth is controlled by using surfactant, which preferentially interact with each different facet. Hexadecytrimethyl bromide, shortly called CTAB, is one typical surfactant for CD-mediated method. CTAB is a cationic surfactant which adsorbed on the surface of gold nanocube in here. The role of CTAB in nanoparticle catalyst is of great interest in both electrochemistry and materials science, because the presence of CTAB contributes to promotion or suppression of chemical reactions for case by case differently. Previously, two studies utilizing electron microscopy based techniques with STEM yields and EFTM reported that the adsorbed CTAB thickness on gold nanorod was about 3 nanometers. So to understand the role of CTAB in nanoparticle catalyst, several questions must be considered seriously. The first is how to remove the adsorbed CTAB ligands from the surface of nanoparticles without causing structural changes. And the second is how to characterize the quantity of CTAB adsorbed rate precisely by utilizing which microscopy techniques. So one other question we wanted to address in this project was how would the characterization be different for ex situ measurement and in situ measurement? So to address those questions effectively, a CTAB removal method was developed to clean CTAB from gold nanocube, which consists of two sequential steps of methanol washing by two steps. First one is methanol washing by soaking the sample for 2 minutes. And the second step is electrochemical cleaning with 2 cycles of cyclic voltammetry in reduction reaction in acidic solution. So this method was also critically important for performing repeatable SECCM measurement. Using 2 microscopies, SEM as ex situ and AFM as in situ measurement, CTAB adsorbate was characterized at the nanoparticles of each step of a CTAB removal method. Gold nanocubes examined in this study has the edge length of 77.25 nanometer and an aspect ratio of 1.03. This slide shows the result of ex situ SEM measurement. Representative electron micrographs of A for drop cast and B for methanol washed and C for electrochemically cleaned, EC cleaned gold nanocubes were shown in this figure. So all SEM images are acquired after co-depositing gold palladium 5 nanometers thin layer. Interestingly, we found that in the SEM image, the square shape of core gold nanocube was distinguishable by the contrast difference as seen in figure A and B. The outer layer is composed of a CTAB organic ligand and conductive coating layer. In the comparison of a figure in A and B, it is clear that methanol washed gold nanocubes show the thinner outer layers with a rougher profile compared to drop cast gold nanocubes. And what we importantly learn from SEM measurement of a methanol washed gold nanocube is that actually CTAB was not detected in FTIR spectroscopy after 2 minutes of a methanol washing, as you can see in the green plot, which is the common macro scale tool for organic surfactants detection in materials science. So this learning suggests that CTAB ligand quantification with only FTIR might have been underestimated in previous studies, which could bring a false assessment for understanding the role of CTAB. Lastly, in EC-cleaned gold nanocube, such a contrast difference of outer layer was not found, indicating that electrochemical cycling effectively removed the CTAB ligands from gold nanocube surface. So the thickness of a CTAB layer was 4.2 nanometer for drop cast and 3.0 nanometer for methanol washed, and it was not detectable in the EC cleaned gold nanocube. Next, in situ AFM topographies of CTAB ligands with single nanoparticles were shown in this slide, which was acquired in deionized water carefully. So figure A to C shows AFM topographies of gold nanocubes, drop cast, methanol washed, EC cleaned gold nanocubes in order. So figure D illustrates the cross-section line profile of the 3 different gold nanocube samples for comparison. So in drop cast gold nanocube, we can recognize a significant amount of CTAB ligands were adhered on the gold nanocube compared to figure B and C. In figure D, with a carton of 77.5 nanometer edge length of gold nanocube located at the center, it is clearly shown that CTAB amount absorbed on the gold nanocubes decreased in the progress of a ligand removal method. So this result is in good agreement with the Situ SEM result in previous slide. The CTAB thickness at the top surface of nanocube was calculated by subtracting the average edge length of nanocubes. So it was 9.9 nanometer for drop cast, 2.1 nanometer, and 1.3 nanometer for methanol washed and EC cleaned, respectively. Lastly, I'm going to talk about correlative measurement and data analysis of in situ AFM and ex situ SEM, which allows us to understand a more detailed level of CTAB ligand layer thickness of individual gold nanocubes. So this correlative data analysis is useful to calculate and compare the CTAB layer thicknesses of in comparison between top surface of nanocube and sidewall, and hydrated versus dehydrated status, and is effective to show the heterogeneity of CTAB absorption on the surface of gold nanocube. So this figure is one example for the data analysis of correlative measurement. So to compare CTAB ligand layer thickness under different imaging environment, as figure A and B shows, the same nanocube was measured by in situ AFM and SEM. So line profiles from AFM, black trace in figure A, and SEM, blue trace in figure B, were extracted, then plotted together as combined in figure C. So the core diameter of this single gold nanocubes edge length can be measured from SEM image as shown in with the yellow dashed line in figure B, where the edge length was 77.8 nanometer. So it means that the height of this nanocube is also 77.8 nanometer. Accordingly, in figure C, a carton of nanocube with 77.8 nanometer within height was illustrated at the center. So in the comparison of the 3 measurements marked in the circular A, B as a full width at half maximum, And so a CTAB ligand layer on the surface of a gold nanocube can be quantified specifically. So at first, in this nanocube, at the top surface of a gold nanocube was calculated as a 2.1 nanometer, which represented the CTAB adsorbate on the hydrated conditions. And also further, the thickness of a CTAB layer in the sidewall can be quantified. So in this case, the CTAB thickness in hydrated, dehydrated, or separately can be calculated. So CTAB thickness as the sidewall in the hydrated status was 21.1 nanometer. In dehydrated condition, the CTAB thickness was 2.6 nanometer. The same correlative measurement and data analysis were carried out for total 8 individual gold nanocubes. This result shows the presence of a heterogeneity in CTAB layer thickness well. The CTAB thickness variation at the top surface under hydrated conditions is in the range of 1.8 to 6.4 nanometer. CTAB thickness variation at the sidewall, as explained in the previous slide, at the sidewall on the hydrated status measured in the range from 13.4 nanometer to 22.3 nanometer for total 7 nanocubes. And in the case of a dehydrated conditions, CTAB thickness was in the range from 0.1 nanometer to 3.9 nanometer. In conclusion, nanoscale CV maps of SECCM with the statistical information reveal that gold nanocubes with the facet 100 have superior hydrogen evolution reaction electrocatalytic activity than gold nano-octahedrons with facet 111. So in situ AFM and ex situ SEM measurement clearly visualized the decreasing trend of CTAB adsorbate on gold nanocubes in the progress of a ligand removal method. And significant amount of CTAB ligands were absorbed after methanol treatment, which was not detected in FTIR spectroscopy. And lastly, correlative measurement and data analysis enabled the thickness quantification and comparison of a CTAB ligand layer in a top versus a sidewall, hydrated versus a dehydrated condition, as revealing the heterogeneity well of CTAB absorption of individual gold nanocubes. Thanks for your attention. I'm happy to answer to your question.